Welcome back. We've got a session now on Pseudomonas infection in PCD, and 4 great speakers to take us through the hour. I'm Michael Lobinger. I'm one of the adult physicians with the PCD service at the Wolverhampton Hospital in London in the UK, and I'll pass on to my co-chair.
Hi, I'm Tamara Paf. I work at the Utrecht Medical Center here in the Netherlands in the pediatric pulmonology department for the PCD service. You're going to introduce a little bit more, or now this session? So it's a very exciting session because we've talked a lot about Pseudomonas in the last sessions, but now there's an entire session. So I am expecting lots of discussions, and I know there are lots of pediatric pulmonologists and adult physicians, so that will be good. I'm very pleased to announce Eric Haarman as the first speaker. He's my PCD mentor, so it's very nice to introduce you. And you're going to talk about the role of Pseudomonas infection and how we can use the registry to get some more information on that. So that's quite exciting. The floor is yours, Eric.
Thank you very much. Thank you very much. Yeah, I'm going to give a brief talk. Within it, a small overview about what is known so far about Pseudomonas in PCD, and we'll make a suggestion for a PCD registry study. As you heard from Kim Nielsen, there's already 3,600 patients in this registry, and I think we can get a lot out of this registry, and this is one of the suggestions for it. And for that, we need you, of course. First, I'd like to start with a study we performed a couple of years ago. We phenotyped the patients in Volendam with the CCDC114 mutation, or now it's called ODET1. And we looked at about 50 to 60 patients, their ENT problems, lung problems, etc. And one of the things we noticed was that the patients who had a Pseudomonas had a steeper decline in lung function during I think we followed up for more than 20 years. Than the patients who did not have a Pseudomonas. And various other studies also show a negative association between the presence of Pseudomonas and lung function, though some of these studies show an association with poor lung function, but not with an increased decline in lung function. So there was always a little bit of discussion, is it now causing the bad lung function, or is it associated with the poor lung function? Instead of a causative problem, which was confirmed by various other studies. Then the next question is, how do you treat patients with a Pseudomonas infection in the airways? And it can be quite heterogeneous. And Susanna Crowley, together with a group of Kim, did a study. They asked various centers, how do you treat patients with a Pseudomonas infection, with chronic infections? And actually, I showed one of the slides in it, a table, which shows the combinations of the various antibiotics you can give, the fluoxetine or no fluoxetine, combined with inhaled colistin or tobramycin. And the most often used, there's a point in here, I think. Well, no, we'll not do that. But the most commonly used combination is the combination of ciproxin with colistin, about 43% of the cases. However, it was well noted that it is very heterogeneous how people, sometimes even if patients, physicians treat the patients and how they treat it. Group in Canada, they tried an eradication regime which was copied from the CF patients, which consisted of patients with new Pseudomonas infections. If they had symptoms, they were treated intravenously, and if they were asymptomatic, with tobramycin inhalation. If you didn't get an eradication, they repeated in on tobramycin inhalation medication, and if that didn't succeed, they gave the patient an intravenous course. And this slide shows the efficacy of this regime. Actually, I do need a pointer a little. This one, yeah. What you see about after culture directly taken after eradication regime, about 74% of the Pseudomonas patients became negative. The patients who did not become negative and you repeat the inhalation medication actually only eradicate about 14%. And then if you give the patient intravenous medication, almost 100% is eradicated. But is it really eradicated? Well, in the long term, this graph shows this, that if you look a couple of years further, about 50 to 60% of the patients remain negative, or otherwise put 40 to 50% of the patients will become positive again. Well, then was thinking, what have we learned from the non-CF bronchiectasis and the CF patients? And this meta-analysis supervised by James Chalmers, combined all the, many studies with an eradication regime for Pseudomonas. And just to summarize it, it's really quite heterogeneous which treatment regime was used, like oral fluoxetine, combination intravenous treatment, combination inhalation medication. But overall, after a year, only an eradication rate of 40% was found. Well, then I looked at the CF, what they do in the CF. So I looked at the Cochrane library, and of course, many treatment regimes have been tested, most of them against each other. I was especially interested in placebo-controlled trials, and there are fewer of these. What you see is that you have an odds ratio of about 0.06, but when you look at the real data, you find after an eradication regime, generally with tobramycin 1 month monotherapy, you find about an eradication percentage of 81% directly thereafter, after 2 months, I mean, and after, I have to correct myself, after 1 month of 81%, and after 2 months, 75%. So we know from CF that it is relatively effective. But actually, this is all that is known about the Pseudomonas and PCD. And I think we have to do two things. Well, first of all, I think we have to make an inventory. What are we doing? What are we currently doing? And more importantly, what's the effect of what we're doing? So I would like to propose a PCD registry study describing the incidence and prevalence of the Pseudomonas across all the age groups, and characterize all the treatments that are given, and more importantly, what's the effect of the treatment? Meaning, will the cultures afterwards become negative and remain negative? Well, it's a multi-center study. I think we need as many people would like to participate in this study from the whole PCD registry community. And in a way, we have to keep it as simple as possible. I did some work in my own center, and even then I found it quite difficult to really extrapolate the treatment the patient was given. Was it negative? Did it remain negative? So in order to make this study successful, it will take some effort, and we have to keep it as simple as possible. And I think we'll first start with the Pseudomonas negativity after 3 months, something like that. More importantly, also as was pointed out by Kim, we really need to do trials afterwards. I mean, we can describe a lot what we're doing now, but in the end, we need trials. And this is something we can also discuss. What kind of RCT will you be doing? Will you do a placebo-controlled study, which is in a way, of course, unethical? In the end, the patients have to be treated. Or do you use a stepped wedge design? Or just do you want to do non-inferiority studies comparing the various eradication regimes? My suggestion would be to do an eradication study with a stepped wedge design, and then I'll wait for these results. But first, I would like to suggest this study. I will prepare a concept sheet, and that will be circulated. And I hope that many of you will participate in this study. Thank you very much. Are there any questions? Yes, please.
Do you use the tobramycin inhaler, the powder inhaler?
Yeah. Well, these studies which I just showed, those were the nebulizations. Yeah. And I think, not sure what... my patient rarely use these tobramycin inhalators, just the nebulizations. Don't know whether any of you guys have experience with that in CF. Kim?
Yeah, thank you, Eric. The problem with such a registry study is actually to know whether treatment data is registered, because it's very, very difficult. And if you think about going back to EPIC and try to see what is described, how long was the treatment and what was given, and so it's a nightmare. I don't want to persuade you to not do it, but I would rather think about a prospective way, some kind of controlled study where you really ask people to do it in a standardized way. The way they may want to do it, but standardize and really always register what they did, what was happening. Like Bruner's profile study also was some sort of standardized way to collect data prospectively.
Yeah, I also see some minor problems. Nonetheless, we will try. And I think nowadays with EPIC, it also has just the advantage that everything you give is really registered. And previously, when we didn't have the digital patient file, we just made the prescriptions using the papers. And then it's very difficult to find out what the patient exactly had and how long they have had it. Nonetheless, I think, well, first we will try in our center, and of course everybody would like to participate. And I think also for the prospective part of the study, I think it's important that people really document it very well, because otherwise we will never find out.
So one of the things that might be important, though, is firstly to know whether it's mucoid, whether it's non-mucoid, the sensitivities. That's going to be fundamental, because certainly we may as well learn for everything we know for cystic fibrosis. And the other thing, I suppose, is also carriage within the nose, because that actually might be really important, that if they've got mucoid Pseudomonas in the nose, you might be getting lots of positive or variable samples.
Yeah.
So maybe that's also important.
Yeah, I completely agree. We should document it well.
Eric, I really like that you are doing that. I think we were asking you for a long time, to start such an endeavor, and I think it's really a tough study, but I think it's important, because we talk very often and long about Pseudomonas, and I think there are many things. For example, we do not know yet what is the presence of Pseudomonas infection. So because we have now so many patients and characterized patients, and with genotypes, we can even find out, is there a difference between the patients with more severe genotypes, less severe genotypes. And I think even this data already, what is presently there, is really important. And then, because this data is present, then you can either go and see what did you do in a standardized way, or what will you now do with what you have found. I think that's really, really good. And I think all the stuff that you would like to answer, I mean, it can be put in the registry, and I think it's good. So I like that, but I completely agree with Kim. Try to reduce the complexity of the study, I think, will be the most important bit. And probably to see whether people are interested, for example, even to collect these samples, and whether they want to study it in more detail, whether there are other microbiologist interests, so that we can cross-link each other. Yeah, so possibly some people now after the talk come to you and say, oh, I can offer this and this.
Thank you very much, Haarman.
I would blow into Kim's horn. Like, if you now do a study, even if every treatment is noted down in EPIC, there will be a huge amount of bias, because the patients who get the treatment in an observational study retrospectively are not the same who don't get it. So if you can at all afford it, I would do a randomized trial, and then you get an answer to which you can trust. I know it's much more work. I know about all the ethics and all the forms and so on to fill in, but I really doubt, if you have data, you can analyze them and make a publication, but really to get robust data upon which...
Yeah, in the end, we really need the trials. I completely agree.
It's probably worth mentioning, actually, that not specifically with PCD, but in the bronchiectasis population in the UK, we are about to start an NIHR-funded prospective Pseudomonas eradication trial with patients randomized either to normal care or for eradication, which obviously, touches on some of the kind of ethics with regards to it, with an eradication regimen being a systemic and antibiotic, which can be oral or IV, followed by 3 months of colomycin, and the other arm not specifically eradicating, but obviously if a patient's ill, then treating as you would treat. So not, as I said, specifically in a PCD population, but equally not excluding a PCD population.
Yeah. Seems like we really would like to see the protocol. Is it already published?
No, the protocol isn't published, no.
Okay.
But it will be, I'm sure it's on, it's called Escape.
Okay. Thanks very much.
Thank you.
Okay, thank you very much.
Great, thank you. So we'll move on to Helle Kroke Johansen, who is from the Department of Clinical Microbiology in Copenhagen. And she's going to tell us about the genetic analysis of Pseudomonas in PCD. The floor is yours.
Yeah, thank you very much for inviting me to present our data. And the genetic analysis of Pseudomonas aeruginosa in PCD, that's of course whole genome sequencing of Pseudomonas aeruginosa. We have already heard some interesting things about Pseudomonas today, new guidelines where I saw a lot of pulmonologists and unfortunately no clinical microbiologists earlier today. I also heard now very interesting things from Eric Haarman presented just before. I have no conflicts of interest to declare. Let's see. Yeah, so from my research and as a clinical microbiologist for years, I know a lot about Pseudomonas aeruginosa. Pseudomonas aeruginosa is an environmental bacteria. It thrives very well in the environment. It can be found in soil and in water and in sufficiently chlorinated swimming pools. And it is able to also infect susceptible people, like people with primary celiac dyskinesia, with cystic fibrosis, COPD, and so forth. But Pseudomonas is able to manage the challenges in the environment. It's also able to manage the challenges that it's facing with when it enters the airways of, for instance, a person with PCD. When Pseudomonas first come into the lungs, they are naive. And naive means that they grow fast, that they're antibiotic susceptible, and that they are able to produce a lot of virulence factors. In the airways, they kind of change the way they behave. Here they have to compete with the microbiota, all the other bacteria in the airways. It also have to be able to cope with the immune system, both the antibodies and the cells. And if I culture them in my lab, I of course tell clinicians, please start antibiotic therapy. And usually they are susceptible, but it's not every time that we are really successfully eradicating Pseudomonas from the lungs, despite they are susceptible to, we think, relevant antibiotic therapy. So what is happening in the airways of, for instance, people with PCD? We know that Pseudomonas is able to mutate, and they create what we call patoadaptive mutations. These are mutations in genes that are more frequently mutated than what would be expected. It's sometimes antibiotic resistance. It can also be the cell envelope, and it can also be biofilm mutations. So the patoadaptive mutations is one of a multitude of armory that Pseudomonas is able to come up with. They can also suppress their virulence. For instance, they go from being motile to non-motile. They can also metabolically adapt, which means that the nutrient that they need can differ with the environment that also changes over the infection time. They are also able to cope with the immune response. And most important is that they are able to grow slowly. And what does growth mean? Growth mean that if you are going to treat a bacterium with beta-lactams, which you have all heard about, then you need a bacteria that have a certain growth rate. If bacteria is not dividing, you are resistant to beta-lactams. And then, of course, we also know from our cell culture models, our air-liquid interface model system, that Pseudomonas is able to penetrate into the tissue and there hide from the antibiotics that are being given. Then they can grow in aggregate or biofilms. And also we have persister cells. Persisters is not rooted genetically, it's a phenotype. So in 2004, I started to systematically collect Pseudomonas bacteria from people with cystic fibrosis. And in 2012, we whole genome sequenced approximately 500 of these isolates, longitudinally collected from approximately 40 patients. And what we got out of that was that we got to know a lot about Pseudomonas from CF. We knew how their behavior was. We knew about clone types. We also found out that we have transmission between patients in our clinic, and we also knew about evolution and adaptation. So I also found out that Pseudomonas, they were persistent in the airways. Before 2007, we thought that Pseudomonas, they were all different. But it turned out that they're not all different. They have clone types, and certain clone types, they infect people with cystic fibrosis. And we found, based on our collection of the almost 500 Pseudomonas isolates from the 40 patients, that if we look longitudinally, that approximately 40% of those children and young adults who got their first Pseudomonas, they also continued as persistently infected, which means that the next time we cultured the Pseudomonas, it was exactly the same as before, even though we had been treated with relevant antibiotics. So it persisted somewhere, either in the airways, in the lower airways, or in the sinuses. So I decided to apply same strategy to people with PCD. So I started back in 2017 to collect systematically and longitudinally isolates from the PCD clinic at my hospital. And of course, I heard before you had a conversation about should we collect 1 or 4 samples. And I must say I'm very, very happy that we have longitudinal collection of our patients' samples, because we know much more about their microbiology, and we know how much better handle infections in PCD. So in Copenhagen, we follow 141 people with PCD. And from those, 65 had a Pseudomonas during the time period from 2017 till 24. And I then collected almost 300 of these isolates, and they were full genome sequenced. And on average, I sequenced 4.5 isolate per person, but of course there were a few with only 1 isolate. I hope that they may come again, I don't know, but they were sequenced. And in some, I had up to 13 isolates during this time period. And the maximum span of years is 8 years, as you can easily calculate. And the average age of the people with PCD at their first Pseudomonas was 26 years, with a range from 3 till 69 years. And this is, of course, not their first Pseudomonas, because I was not in the clinic when many of these patients were seen initially in the PCD clinic. But this is as good as we can do. And for some people, it's the first one. For some, it's maybe their isolate number 50. I don't know. And the cohort of people with CF, that was much more well controlled because it was the first isolate and subsequent isolate. But here, this is the best we can do until now. So what did we find out about persistence of Pseudomonas aeruginosa in the PCD airways? We found out that 44 patients with PCD that had Pseudomonas in 1 year longitudinally. 40 of those 44 had exact the same clone type, which means that we have persistence of same clone type in 90% of these people. And if we move on and see how many people have had continuously culture of Pseudomonas for 3 years, that was 34. We found out that 30 of these 34 had exact the same clone type, which is 88%. And then we looked into how many of the initial patients had Pseudomonas continuously cultured over a period of 5 years, and out of those 28, 23 had exact the same clone type. This is a lot of persistency. So when Pseudomonas is in the lungs of people with PCD, they persist. So we also looked into, number one phenotype to stay in the airways of people with PCD and also with CF. So on the left-hand side, you will see what we found from the genotyped and phenotypically characterized Pseudomonas isolates from CF patients, that we see Pseudomonas is gradually having a slower growth over the years. If we go to the right-hand side and look into the only 223 isolates that we successfully cultured and measured growth rate, you see that if we compare to the wild type P01 with the purple dotted line, this is the average growth rate of a wild type bacterium. You see that almost all of the PCD isolates are below the line, which means that over time, and also in patients, these Pseudomonas bacteria, they grow slowly. And therefore, maybe also a reason that we are not able to eradicate these bacteria. And this is just in clone types that are more frequently found in the population of people with PCD compared to others where we have enough bacteria to really draw a line. And in these different clone types, 8 clone types, you see that they all decline over time in their growth rate. So a very, very good way of surviving in the airways and how not to be eradicated by antibiotics. This is a more busy slide, but it's biofilm formation in isolates from CF patients, and again based on our almost 500 isolates. And if you look on the left-hand side, these are biofilm measurements in a microtiter plate in the 2 first years. And the blue lines below the line is the wild type, those that do not form very much biofilm. And the red one are the evolved ones, those that form biofilms. And if you look on the right-hand side, you will see that there are many more red lines going above the line, which means that biofilm formation is a characteristic that develop over time, and also a survival strategy. So if we go into the isolates from the PCD people, 223 isolates, again, I have inserted the wild type P01 as a dotted purple line. And you also see that over time, exact same characteristic, biofilm formation is developing in isolates that are evolved. So to conclude, I think that we have exact same situation in people with PCD as we see in CF. The growth rate decline exactly as we see in CF. And evolved isolates in PCD, they form biofilms exactly as what we see in CF. So these findings, they of course, and I hope sincerely that you will all think about this when you are in the clinic, because this will have implications for how you treat patients. Also early eradication, because they stay in the airways of people with PCD if you do not eradicate. Also, microbial diagnostics should not be done once a year, but at least 3 times a year, so we are completely certain what is going on in the airways. And so that we do not miss the opportunity, the window of opportunity to eradicate Pseudomonas. Of course, also infection control, because we haven't seen transmission among the isolates that we have sequenced, but transmission have been seen and reported at several occasions in CF. And of course, also, this must definitely influence guidelines in the future for PCD. And the project group with people from Copenhagen, and many thanks for listening.
So we have very short time for questions, so 1 or 2. Joel.
Hi, thanks for the data and talk. I was wondering if in some of the patients there were multiple genotypes present, and if that was correlated to any clinical outcome, if you could say anything about that.
I haven't had the opportunity to look into clinic. I only had microbiology, so I'm sorry.
And were there many with multiple genotypes?
A few. I think it's the same what we see in CF, that one clone type come in and is outcompeted by the another one that stay in the airways. So most likely same pattern in PCD.
Thanks.
Hi, on a similar vein. The longitudinal data you described was on people who had not had eradication, am I correct?
Yeah.
So you described quite a high degree of persistence of the same genotype coming back. As you can see, my obvious question is, so in the people who did have eradication, I'm aware that certainly on the CF world in Copenhagen you do a lot of eradication, did they come back the same genotype or was it a different clone?
I'm sorry, we haven't looked into it. We ended this study on Friday last week, so I haven't had the opportunity, but I'll see which clone types they have.
Okay, thank you very much. Hot off the press. Thank you.
Thank you.
So moving on now, we've got a talk entitled Patient Perspective on Antimicrobial Resistance in collaboration with AMRlung CRC from Dr. Papadopoulou from Greece.
Well, thank you for the invitation. I'm a clinical pulmonologist, adult pulmonologist in Greece, and I'm here as a member of the AMRlung Clinical Research Collaboration to present to you this data. These are not specific to Pseudomonas infection, and we only recently have been able to explore patient perspectives on antimicrobial resistance in a patient survey, which was created by, why isn't this working? How to change the slide? Okay, thank you. Thank you. In this patient survey, which was created by members of the AMRlung CRC Clinical Research Collaboration, chaired by Dr. Anand Shah, Mikkel Ekelenkamp, and Stephanie Te, who is also here today, and also from patient representatives from the European Lung Foundation. So this patient survey was translated and held online in 21 languages over a 2-month period in 2024, and collected over 2,000 responses across 57 countries around the globe as shown in the map. So respondents had a range of chronic respiratory diseases, most commonly bronchiectasis, cystic fibrosis, and asthma, with primary ciliary dyskinesia in about 7% of respondents. This presentation will focus on this population, the data from this population, but of course you can have a look at the overall survey findings in the early view publication, which is available in the ERJ Open Research. So we had 139 respondents with primary ciliary dyskinesia from 17 countries. Most were from the United Kingdom, and followed by France, Germany, and the United Arab Emirates. So most were from high-income countries, and there were only 5 participants from middle-income countries, with none from low-income countries, which is of course expected due to higher diagnostic services for primary ciliary dyskinesia in high-income countries. Most participants were in the age group of 25 to 34 years of age. Females, women are generally more likely to respond to surveys, and this was also the case here, with 7 out of 10 participants being female. And most respondents had attained a higher education level with a bachelor's or master's degree. Also, 60% of respondents were followed up at specialist national centers, as expected, of course, with primary ciliary dyskinesia, but we also had an amount of respondents who were treated at local or regional hospitals or even with family doctors. And there was a significant 12-month respiratory-related burden, as 4 out of 10 respondents had hospital admission or other emergency healthcare contact within the last 12 months. Most were had been diagnosed for more than 10 years. And apart from primary ciliary dyskinesia, they had at about 60% also comorbidities, most commonly sinusitis, cardiovascular disease, or gastroesophageal reflux disease, which is of course expected for patients with primary ciliary dyskinesia. And some of them also received long-term immunosuppressive medications, corticosteroids. We don't know the reason for that. So, to the findings. Prophylactic antimicrobials were prescribed for long-term use to about half of the respondents, and in the wider patient survey, multivariable logistic regression models showed that patients who had been using prophylactic antimicrobials were more likely to recall AMR-associated infection, which was here the case in 36% of respondents. But as you can see in the graph with the dark gray color, about 17%, which is a significant amount, were not sure. So we did have some recall bias here, definitely, and this percentage might be even more high. And specifically regarding their last antimicrobial course, it was alarming that 15% perceived antimicrobials as ineffective or only a little effective, although almost half of the respondents received a prolonged antimicrobial course, and more than 30% of respondents had no sample sent for culture or sensitivity testing prior to receiving their last antimicrobial course, and that despite the high infection-related burden. Also, a significant number of respondents, at 23%, occasionally used antimicrobials without medical prescription. This was a self-reported. Self-reported data, so we haven't looked at their medical records, but patients reported that they occasionally used them as leftover from previous times or directly over the counter, which is of course alarming considering the high infection-related burden that these patients face. And in the wider patient survey, multivariable logistic regression models showed that there was higher likelihood for self-medication of antimicrobials if patients perceived physician prescriptions as delayed. And this was the case here among PCD patients in 20% of the cases, which was twice as high as the wider patient group, where it was only 9.5%. So among those who received prophylactic long-term antimicrobials, the burden of antimicrobial use remained still high, with 50% of them also needing intravenous courses during the within the last year. And as you can see with the dark pink colors, more than half of them also received more than 2 courses of intravenous antimicrobials within the last year. The majority, more than 50%, at about 60% of respondents, report that they had a moderate to excellent understanding of antimicrobial resistance even before participating to this survey. And they reported that their main information source regarding AMR was their interaction with their healthcare professionals, but also hospital or government information, and of course, patient associations. Regarding specific AMR-related pathogens, as you can see, Pseudomonas aeruginosa was by far the most identified related pathogen by patients, and then followed by MRSA, but a significant number of respondents did not know specific pathogens related to AMR. But they did identify specific antimicrobial resistance contributors to antimicrobial resistance, specifically antimicrobials overuse, incorrect dosing or duration, and also antimicrobials used in animals, more than 40 patients. Participants, more than 9 out of 10 actually agreed on the significance of raising AMR awareness among patients, on the significance, and they would also like to understand how antimicrobial resistance may impact on their future health, how to identify signs of bacterial infection, ways to prevent antimicrobial resistance, and why it is important to avoid unnecessary antimicrobial use, but also more specific practical issues, such as how often they should be tested for AMR, and what could the vaccination impact on AMR be. And they also viewed AMR as a significant threat, both on an individual and global level, and expressed concerns regarding the related ineffectiveness of antimicrobials, AMR's potential impact on medical procedures, and also AMR transmission between patients or from patients to their family. And they valued research areas regarding how to distinguish better and differentiate bacterial from other infections, research on novel antimicrobial types, and research on ways to prevent resistant pathogens transmission between individuals or from the environment. And regarding the preventive strategies against AMR, more than half of the participants valued a better ability to identified or recognize as a preventive strategy a better ability to identify and treat infection. So in comparison to other patient groups in the wider patient survey, participants who had primary ciliary dyskinesia and cystic fibrosis, as you can see, had a higher likelihood to recall AMR-associated infection, but they were also more aware than other patient groups about AMR. And of course, overall, these data were captured in a survey with voluntary participation, so they are subject to potential recall bias and also selection bias, but they do definitely suggest an increased burden of AMR among patients with primary ciliary dyskinesia, as expected, of course, and the need to improve and find strategies and policy to tackle this area. These are not specific to Pseudomonas infection again, of course, but they are an important step to understanding the patient perspective on AMR burden. So we have to thank, of course, all survey participants and the national patient organizations for the survey promotion, and of course, members from the AMR Lung who participate in the translation and dissemination of the survey. Thank you.
Thanks very much indeed. So time for a question or two.
Thank you very much for this presentation. I find it very interesting because in our recent patient survey with ELF on PCD patients about research priorities, one of the unique priorities that the patients identified that was not identified by healthcare professionals were that they wanted to know more about antimicrobial resistance and about effects of long-term use of antibiotics. So I was wondering if after this work you plan to do any educational material for patients through the CRC, because I think this would be very relevant also for the PCD population.
Yes, there are also definitely plans for next step. We plan to have some qualitative research also to pinpoint specific areas while interacting with patients. There will be some educational materials also in the ELF site. also with regards to the survey results. But we haven't yet organized anything specific to that. But thank you for the suggestion.
The selection bias is difficult, isn't it? Because obviously if you ask people if they want to do a survey on AMR, then the people that say, oh yes, I'll do that survey, are likely to have some knowledge of it and some views about it as well. And I suppose also highlighted by the very high percentages of long-term antibiotics and things like that. Obviously the data was quite skewed towards a particular group of adults in the UK in the PCD population. Was that the same in other conditions as well, or was it specifically PCD that was so sort of demographically uniform?
Well, adults were definitely participated at a higher percentage. We gave the opportunity to caregivers to also respond of participants to include ages under 18, for example. We didn't have a look at specific countries and ages within each chronic lung disease, but we did include high income versus middle income countries in the analysis, in the multivariable analysis, in the wider patient survey, but we haven't had a look at the specific data in all chronic lung diseases. And we only did this analysis for this specific presentation here as well.
Because I suppose it's difficult, isn't it? Whereas if you're capturing a specific characteristic or behaviour of a PCD population, or actually just representing, the UK Health Service with regards to access to antibiotics and those kind of things. But I'll tease out, I suppose, when you look at the others. Thank you very much indeed.
Thank you very much.
Okay, so the final talk on this section is Maretta Long from Dundee, who's going to be talking about new therapies against Pseudomonas infection. Thank you.
So thank you very much for the opportunity to join the meeting today and to present. It's my first PCD meeting, so I'm very excited to be here. You may recognise this slide from about 20 minutes ago. And it's a really nice figure from Professor Johansen's review that she has already presented. So I won't go too much into this, but what I really wanted to emphasise from all of the mechanisms that we know that Pseudomonas uses to persist, that these are quite reliant on evading the immune system. So we talk a lot about antimicrobial resistance, but Pseudomonas is actually really good at avoiding the body's own natural defences and has all these kind of different mechanisms to be able to do that. So it's actually a really quite smart bacteria. These are all the mechanisms that it does use to evade antibiotic treatment success. So it has several innate mechanisms, including things like low membrane permeability and the ability to acquire resistance, as was already discussed earlier in the session. So we do need novel therapies to treat Pseudomonas infection. We know that it's a prominent problem, and there are several of these that are in development currently. So things like vaccines could be of interest, and these are currently, I think, in quite early stages of development, but could be developed against targets on the Pseudomonas surface, for example. Novel antibiotics are in development and have been trialled in some cases, although I think in the interest of AMR, that's probably not going to be the main focus of kind of novel research. We could use things to enhance the body's own immune defenses, antimicrobial peptides, for example, anti-inflammatory treatments. Phage or bacteriophage is, of course, of interest quite recently, and there have been small studies and case reports of largely compassionate use of Pseudomonas targeting phage, either cocktails or single phage therapy, which have had some success, but do need to be explored a little bit more in a little bit more detail in larger cohorts of patients. But the therapy that I'd really like to talk about today and that we've been researching is monoclonal antibody therapy to target Pseudomonas infection in those with chronic infection in the airways. So I think this has already been emphasised in the session, but we know that Pseudomonas infection is common in bronchiectasis in particular, with around a quarter of patients, at least in Europe, being infected with the bacteria. We know that it's associated with disease severity and adverse outcomes, hospitalization, survival. And what we also know in bronchiectasis is that the immune system might not be as efficient at clearing the bacteria. So in those with more mild and severe disease, there seems to be a defect in neutrophil phagocytosis of Pseudomonas, which again helps the bacteria to further persist. So besides all of its own mechanisms to kind of evade the immune system, neutrophils seem to be less able to actually do their job as well. So all of this compounds into quite a nice environment for Pseudomonas to persist and become a chronic infection. So we've been studying a monoclonal antibody called gromubimab, and this has two targets on the surface of Pseudomonas aeruginosa, and this is PCRV, which is part of the type 3 secretion system. So this is the molecular syringe by which Pseudomonas can inject its toxins and virulence factors into host cells and epithelial cells. The second target is PSL. So this is an exopolysaccharide, which can do things like promote immune evasion, so it prevents complement deposition onto the surface of the bacteria, as well as contributing to biofilm formation. And so by blocking these two targets on the surface of Pseudomonas, we think that we could achieve both greater host recognition of the bacteria as well as reducing bacterial virulence. So we performed a proof of concept study initially in the lab to understand the potential for this treatment in bronchiectasis. And the results of this study are now published last year. And what we showed was that the two targets are quite prevalent in a global collection of Pseudomonas isolates from bronchiectasis patients. So 99% of individuals that we looked at had PCRV, and this is by whole genome sequencing, and around 73% had PSL, so slightly less, but overall 99% expressed at least one of these two targets. We also showed that when gromubimab is present and we use primary isolates from patient airway samples, we could reduce epithelial cytotoxicity of the bacteria, as well as reducing mortality in a murine model of Pseudomonas infection. And finally, we showed that when gromubimab was utilized, we could actually improve the killing of Pseudomonas by patient neutrophils, and that there was no difference in response if we look at healthy individuals or bronchiectasis patients. So this was all really encouraging and led us to the design of the Great True trial. So this is a phase 2 proof of concept trial for gromubimab in people with chronic Pseudomonas infection and bronchiectasis. So patients had to have CT-confirmed disease and be culture positive or PCR positive for Pseudomonas in a sputum sample. Patients received 12 weeks of treatment. So there were 3 potential arms: a higher dose of gromubimab, a lower dose, or placebo. They received treatment by intravenous infusion every 4 weeks for the 12-week period, and we also had a 12-week off-treatment follow-up period. The primary endpoint for the study was bacterial load assessed by quantitative culture at the end of the treatment period at week 12. But a key secondary endpoint was looking at the change in bacterial load over time, as well as various other endpoints, including quality of life. So this was a relatively small study. We had 12 people in the high dose arm, 13 in the low dose, and 12 in the placebo group. And in terms of patient characteristics, this was relatively well representative of a kind of bronchiectasis cohort within Europe, and the groups were relatively well matched. So we were really pleased to see that the trial met the primary endpoint. So as a proof of concept study, we used a one-sided p-value to assess statistical significance. And we saw a significant reduction in bacterial load at the end of the trial at day 84 for the lower dose of 500 milligrams of gromubimab. And we also saw a trend towards reduction in the higher dose arm as well. And this is probably equivalent to that achieved with successful antibiotic treatment. So it's a reduction of around 94% in bacterial loads. One thing that differs quite significantly from antibiotic treatment is that patients in both arms reported really significant improvements in quality of life. So we consider the minimum clinically meaningful difference in the St. George's Respiratory Questionnaire to be around a reduction of 4 points, where patients actually really feel better. And we are able to achieve a 10 to 12 point reduction for both treatment arms here. So we think this is really quite important, and patients are saying that they feel better. And we've used multiple quality of life measures, including the bronchiectasis impact measure, which all tend to show the same thing and perhaps a little better for the higher dose arm in terms of things like cough, sputum, and how patients report that the disease impacts on their daily activities. We also saw that there seemed to be a prolongation of time to first exacerbation, and both of these trial arms, but particularly for the higher dose arm, there was 83% of patients remained exacerbation-free at the end of the treatment period at week 12. And our lab works on neutrophils, so we wanted to understand if this actually impacted lung inflammation. And we see a really significant reduction in multiple neutrophil granule proteins that we know are both disease biomarkers and that are important in disease pathology, including azurosidine-1, myeloperoxidase, and neutrophil elastase. And again, this was more prominent in the higher dose arm. And although this was variable over time, we can see at the end of the treatment period, this is really at the kind of lowest that it can be. We also looked at levels of airway mucins, and we know, of course, these contribute to things like sputum viscosity, and again are linked with more severe disease. And we saw a significant reduction, particularly for the lower dose arm for MUC5AC, as well as MUC5B. And when we look at the ratio of these two mucins, we see that that's also a significant reduction for the higher dose arm as well. So to summarize, we have shown that in people with bronchiectasis and chronic Pseudomonas infection, gromobimab successfully reduced Pseudomonas bacterial loads, and this met the trial primary endpoint. Gromobimab at both doses showed significant clinically meaningful improvements in patient-reported quality of life. Time to first exacerbation was also prolonged with one of the doses of gromobimab used. Sputum markers of inflammation were reduced, as well as mucins at the end of the treatment period. I haven't shown the data here, but the safety of gromobimab treatment was similar to that for placebo treatment. And relevant to the mucin, I think utility in PCD may certainly be of interest. We had one person with PCD in the study. And so hopefully larger studies of gromobimab will happen and will include PCD patients. So yeah, I'd like to thank everyone involved in the GREAT2 trial and in the proof of concept study. This was done in Professor James Chalmers' lab at the University of Dundee. We had multiple funders for the trial, including AstraZeneca, ERS. And I'd really like to thank all the participants that took part in this trial and allowed us to do this research. Thank you.
Thanks very much. Time for one or two questions before the next session. I'll ask one question then. I mean, it was quite striking to have such sort of positive secondary endpoints, particularly things like the patient-related outcome measures, the exacerbation measures, where typically antibiotic studies in Pseudomonas haven't had such success in those areas, despite similar reductions in CFUs and amount of Pseudomonas. Do you have sort of thoughts on why the secondary outcomes were so good with this approach?
Yeah, so it was one of the things that was really interesting, and achieving kind of, same rates of bacterial reduction, I think, gives us quite a key insight into the mechanism of benefit. So we achieve really quite significant reductions and changes in airway inflammation. We know that this is quite strongly driving disease severity in bronchiectasis. Of course, neutrophil elastase is a therapeutic target, and with TPP1 inhibitors, we know that provides a really significant benefit beyond what antibiotics have also achieved. So we really suspect that modulating inflammation is actually more important than reducing the bacterial load itself. And it has a kind of dual role of enhancing the immune response, but also reducing epithelial cell death, reducing airway inflammation. So I think those kind of multiple modalities that antibiotics don't achieve are probably driving the benefits that we see.
And do you not think that, but I mean, I don't know if it's been measured, all of the azurecidin and things like that during trials with inhaled antibiotics, for example. Would you not expect them to have similar reductions in inflammatory markers due to reduction in bacterial load, or you think this is actually having an additive anti-inflammatory impact, maybe?
Yeah, so I think some studies have certainly shown a reduction in neutrophil elastase. I'm not sure if azurecidin has been measured since it's kind of more recent biomarker that we've been interested in. But I think in a kind of larger, broader inflammation that is happening in bronchiectasis, as much as we focus on neutrophils, they're not the only important cell. So I think the kind of broad immunomodulatory effect is probably much more important and something potentially not achieved by antibiotics, because you're also targeting the virulence as well as kind of clearance.
Thanks so much for this exciting data. We're going to wrap up this session. So we learned we need to be frequently sampling, we need to be fast in eradication, and we also need to look at immunomodulation, I think, to summarize the session. We're going to move to the international updates from all the collaborations. So I'm going to give the microphone to Myrona and Kim, I believe, to tell us everything about the new updates you have.
I hope you still have a bit of energy left for this last session. We have a small change in the schedule. So I swap positions with Hannah Mitchison, who will talk first to present an update on the ClinGen project. And then we will continue with the program as it is.
Okay. Yes, so thank you very much for the invitation to speak today. I'm really happy to be representing the Work Package 2 group from BEAT-PCD. This is a work package to improve the genetic interpretation of PCD, and one of the main focuses is development of the CiliaVar database, which will be the first open access variant resource for PCD. I'm not going to talk about that. I've been asked to talk about the ClinGen gene collaborative project that Work Package 2 has been engaged in, which was really instigated by Suzanne Crowley, who's in the room. So just a couple of intro slides. We've actually heard quite a lot today already about the challenges of PCD genetic diagnosis. And you've heard this stat, which seems to have been the same stat that since my whole time, the last 20 years of PCD, or perhaps that's a bit in the last 5 years, should I say, that even now we have up to a third of PCD patients who leave genetic testing without a complete diagnosis. So there's a list of issues why that would be. We have multiple genes associated, up to 70, with variable evidence of their link to PCD. We have large numbers of variants that are so incredibly rare, they're actually 60% unique to individual families. That really complicates whether they're going to cause disease or not because of lack of evidence and backup. Unless they're really clear-cut bond or variants. Many variants of uncertain significance, or VUSs. We have a very specific problem that actually, if you read this recent reference from Fleming et al., the hide-in-2 problem, that's being actively tackled, but it remains the case that clinical centres quite often don't even look at hide-in, because it's a complicated gene with an almost exact copy on a different chromosome that's nothing to do with PCD, and it really complicates because the sequences are almost identical. So the Fleming et al. Showed that it's actually quite a common gene causing PCD, so it should be certainly considered. We have the complexity of cilia gene biology, which means that our understanding about the relationships to phenotype is pretty has a long way to go, and that complicates the genetic interpretations. We've heard today how important it is when you find a gene variant to know that it's correct, it's causing the expected biology and the expected structural defect and so on, but we really don't understand everything about that. We have many subtle structural changes. So 2 slides about VUS, and actually it was very nice to see Rob. Thank you, Rob. I haven't seen your slide on VUS before. This is more or less the same sort of thing that I'm trying to emphasize here. I don't know if people can see the panel on the left. ClinVar is an NIH, a very important repository. This is basically where everyone puts their variants. If you type in primary ciliary dyskinesia, there are thousands of variants, and there are variants that are likely pathogenic and pathogenic, and there's maybe, looking at it, 4,000 or 5,000 of those, but that's vastly outweighed by VUSs. These are variants where there's just insufficient data to know whether they cause disease or not. For example, they're a point mutation in the DNA that causes a single amino acid swap, or they're an in-frame deletion that doesn't, some of the protein can still be read, or they're deeper intronic variants. These sorts of things, if you don't have multiple patients or if you haven't done functional testing, remain, they're not diagnostic. They are VUSs, and you can see 2,000 conflicting classifications and 10,000 VUSs for PCD in the current version of ClinVar. ClinVar data it's great that there's one tool that people put their variants into. It's collected from publications, from genetic labs, from commercial gene test laboratories. Invitae put all their stuff in, and various other companies who screen patients. So whilst it's a key resource, it's not heavily evidence curated by experts, and we're the experts on that in this very room and beyond in the PCD community. So throwing in many variants that may really not be causing disease. And this is the fantastic paper that came out from Ben Gaston's lab with Hannah. William Hannah was the main author in 2022. So if you look at the top panel here, I won't go into the details, but this is where we get our statistic that we now think that PCD affects globally 1 in 7,500 people. That's the very top line. That's in everybody. Actually, in non-Finnish Europeans, it's more like 1 in 10,000. And that is data where they carefully curated it to take out any VUSs. That is only the pathogenic and likely pathogenics. The bottom table shows the comparison when you include these VUSs, where you do not know if they cause disease. And you can see this statistic goes to 1 in 127 people globally with PCD. So we know that a lot of these VUSs are hanging around and are not disease causing. So work package 2 is all about the need to improve our understanding of the genetic basis of PCD. Genetics is a really unique uplift to diagnostics. And of course, we've heard, and I'm allowed to put this line in from Amelia, that the new diagnostic guidelines that have come out today certainly have pushed genetics way up the tree in the diagnostic pathway. You get a molecular diagnosis, which gives biological understanding of the basis of the disease. When you get a genotype, you can do things like carrier testing, segregation studies. It gives some unique advantages. Genetics is starting to be able to predict the disease course, the severity. We've heard a lot more emerging about that today. Certain variants are going to be causing more severe disease, and some are associated with mild disease typically. We can predict different organs outside the airway, whether they're going to be involved. Syndromic forms are emerging. And of course, absolutely key, you have to know your genotype if you're a patient who wants to go into a genetic therapy trial, such as the one from RECODE. And those are going to be increasing. So this all needs a systematic approach to evaluate the causality of genes and variants that we find in genetic testing. And that's what the ClinGen collaboration that BEAT-PCD has conducted with a very large team of people. This is the GCEP leadership group. So there's also a VCEP, and I'll list those people. It's important to make clear who's done all the work. So ClinGen is an American NIH funded project. It's called the Clinical Genome Resource. It's got significant funding behind it to define the clinical relevance of genes and the variants in those genes for research and precision medicine and trials, etc. So this is gene disease validity curation is what we've been doing. So we BEAT-PCD set up with a ClinGen, the motile ciliopathy GCEP, the gene curation expert panel. So first of all, looking at PCD genes, so not variants at this point, except as part of understanding whether a gene is causal. It's been going a long time with monthly meetings since actually 2021 with a large group of people, experts, clinical experts, the very important biocurators who produce the research into all the different genes, and Christina, our coordinator. And we're curating at the moment 70 genes for PCD, but some of them we've also had to split and curate for things like spermatogenic failure, hydrocephalus, and heterotaxy that we know are also features of some parts of PCD. So the ClinGen gene curation process, we have selected these 70 genes. There's a literature review done by curators per gene, and there's a very strict evidence scoring framework. The classification, they are definitively linked to being PCD genes, PCD causing genes when they're mutated. There's several categories: definitive, strong, moderate, limited, to no disease association, or disputed, refuted. Each of the evidence scoring and the work done by the biocurator is then undergoes expert review within the clinical and researchers within the team. A consensus decision is made about the classification, and there's a public database entry, so it goes online, which is great. The types of evidence are genetic and experimental. So genetic evidence, how many patients have variants in that gene? What does the segregation data look like? What's the functional impact of the variants that are reported? Experimental evidence in animal models, allele cultures, biochemical function of the gene and the gene product, whether it interacts within a known PCD-related pathway, and rescue experiments, pushing the gene back in, does it restore function in knockout models? So just an example, DNH5, still the most commonly mutated globally PCD gene. So this is the table that gets drawn up with the genetic evidence and the experimental evidence, and this is tailored for PCD. So the evidence is based around the things that we've been talking about today, electron microscopy and so on. So you can now go and look and see that DNH5 is definitively linked by the group to PCD, and there's a really nice report summarizing all the evidence. So we've seen this a little bit earlier from Amelia, that this is the status of the GCEP curations in September 2025. So up at the top, you'll see that we've done 74 curations on 60-something genes. So different curations for different diseases, the sperm failure or the PCD. With these, but this is the result, and you can see that for each gene we have the experimental evidence in red and the genetic evidence in blue. So you can see DNH5 way over to the right as this definitive and strongly associated, compared way down to the bottom with, for example, DNH8 with zero evidence. And you can see that these genes, for example, DNH6, the geneticists in the room will be familiar with some of these. There's much more experimental evidence for some of these genes, but there's hardly any patients reported. There's hardly any human mutations. So we've had to give them limited and disputed classification. And DNH8, we know it's not expressed in the lung, it's only expressed in the sperm. Okay, so that's why it is disputed. A survey, this is a slightly separate work, but associated. A survey was sent out via BEAT-PCD to PCD centres to understand how they perform their genetic testing. So many rely on gene panels. 11 centres provided their custom gene panel information, and others use commercial panels. This is the list of many companies that provide gene panels on the left. And you can see that of our curated genes, first of all, we've got highly validated PCD genes are not present on the vast majority. So TUB4B, that's definitively associated with PCD. It's got good evidence, but it's not there yet. On panels, it's too new. And the TP73 is another very important gene. Again, it's only on 40% of panels, whereas our disputed genes, and I'm just going to pick up in the blue, DNH8 again, that is not a PCD gene, definitely. We have disputed it being a PCD gene. That's kind of our most critical rejection. That's on 55% of PCD panels. So these things are important and can really guide. And thanks very much. This is Debbie Morris-Rodendahl's data, who went through all these, the gene panels and did the survey. So yeah, important data. So we've got 74 evidence-based curations so far for 62 genes. We've got about 10 left. We can make recommendations, and you can see them published already, on which genes to include and exclude in the PCD testing panels, which have to be constantly updated. There's a recuration process. So if DNH5 was, or some of the genes have been curated first 4 years ago, so every 3 years or so. And if there's new evidence that arises, there's a recuration to see whether we can update. As a result of the GCEP, we've launched the variant equivalent, so the motile ciliopathy VCEP, working with ClinGen on variant curation. And the two chairs are not here in the room, but I'm hoping they're online. Marie Legendre and Maimouna Zariwala. Really nice. You can see that this is a highly collaborative from Europe to America. And because they're not here, I've just got this one slide, and we have Tom, who's a major rep from the VCEP, several other people in the room. So that was on the back of the success of the GCEP, launched a couple of years ago. The VCEP team has already adapted the ACMG guidelines, the classic guidelines for variant classification, and they've developed a PCD-specific variant classification rules. They've chosen DNH5 as their first example to test those rules because it's the best known gene globally. These rules have been submitted, feedback happened, and they've been finally approved by the Variant Interpretation Committee at ClinGen. So now 50 DNH5 variants have been selected for a classification pilot. You can just imagine what an absolutely astronomically vast project that would really be to curate all the variants in PCD. So we have guidelines and 50 VUS or conflicting pathogenicity interpretation DNH5 variants have been chosen. There's been some mock curations, and now we have this pilot curation ongoing for a systematic looking at the variant causation. Okay, so PCD genetics, as we've heard reflected in the new guidelines, an increasingly essential part of diagnostics. I don't really need to tell anyone, but it's not yet a gold standard test. We certainly don't know all the PCD genes, and we don't know all the variants. The true incidence of PCD is not understood. How many genes there are? Gene discovery continues, VUSs expand, gene panels will need this constant updating. So many challenges, and these are to go to open access ClinGen tools, worked up with the BEAT-PCD expert group to resolve VUSs and enrich our biological understanding. So the GCEP for gene curation to improve variant classification and clinical interpretation, and now the VCEP working to compile the PCD-related variants, supporting research and better geno-pheno correlations. So that's it from me. I want to acknowledge the GCEP again and the whole of work package 2 led by Suzanne, and of course the patients and their families for all this information. Thank you.
Thank you very much, Hannah. Anyone having questions to Hannah?
Yeah, I'm always interested in the classification of variants. So there's 50 variants. So what will be done? Is it like computational models, or are they going to test this in the lab, or is it just going to go through the guidelines?
Yeah, so they have a variant-specific guideline. So I haven't been so closely involved in that. It's going to be a scoring table in a similar fashion, and we'll start to see variants having scoring based on I would say online available evidence. So again, to go through the functional and the genetic support for those. You know, how many patients has the variant been seen in? How does it look in AlphaFold? All these bits of evidence, but it will not be that the biocurators look at the literature, and then as you go along, you build up more information about what makes sense for a variant. But I think that will come out. So I was mostly talking about the gene variant, the gene curation. The variant curation is quite specific, developed within that group.
I have a very practical question, Hannah.
Oh.
Thank you very much. No, I mean, you very nicely showed also from the results of the survey that there are still several centers that they use panels that they're not the most appropriate. And these really valuable resources, they're open source and people can access them. How do you think that we can promote this correct information in the best way so these panels are updated and the correct panels are used?
Yeah. Thank you very much. Well, there's a somewhat overdue publication. We've been doing it for 4 years. We have to, of course, get the results out there. They're not really out there. I'm not sure many people really know about the resource. So there'll certainly be a publication about the nuts and bolts of how we do it, and indeed we'll find out about how the variants are classified as well. And then the idea was to have a opinion piece, some kind of this is what you should include, these are how we did it, how important it is not to be screening patients that we know are not highly associated with the disease, because there's many variants in genes and it can be very confusing. So I guess, and whatever, I mean, thank you very much to the BEAT-PCD for all your channels of advertising these things. Yeah.
Yeah, I think it's a little bit more complicated. It's not like you can tell someone to make this and this beta panel. For example, hydine was not tested in most centers in the US, and this was a medical legal issue because hydine is duplicated. We published that. It's very complicated, and they do not want to write up wrong reports. And very often you need additional tests, so we included this. Thing is that you could do, for example, antibodies like SPEF2 and others and so on, and you can look by RNA. So it's a little bit more complicated. So labs tend to be on the safe side. So they do not want to have litigation issues. So it's really complicated. It's not like the way we can recommend something. So people can read, I mean, what published all the gene. The homepage, I looked a few times and I was confused that something like DNH8 in the beginning was just written there. Now I'm very happy that we just say it's excluded. Yeah, as you know what I mean, it was sometimes not easy for everyone to understand. And I think the other thing is there are genes that cause PCD that are not on the list. So it's not a complete list. And I think it's also important for everyone is that if you find a genetic defect that is not on the list, but it might be published already and it's a PCD gene, it's only not on the list.
Yes. So to be clear, it's an ongoing process. So we have things that are not published yet. We have 72 or something that we are curating. That's based on trawling the literature. So if you have genes that you know about, we'd really love to hear about them, or it's very important to publish them, then we will be aware. But yeah, I agree. This could be going on forever, the constant curation. So it's nice that there's funded staff at ClinGen who can support it. Yeah.
I was just going to make a short suggestion to Myrona's comment, actually. So one thing is that Maymuna's put the classifications in her gene reviews chapter, which is a really useful chapter on the genetics of PCD, and is kind of repeatedly updated, and she's put the classifications in that. Yep. So that's really useful. But also I think we should probably just put a link on the BEAT-PCD website of a really, really simple list of the genes and the classification that would be available to patients as well as to clinicians. So it's really, and just keep updating that so that, you know, without all of the background evidence, just for something that's really simple. And also to say, I don't think people do always play it safe, because we get loads of referrals of people who are diagnosed with PCD who don't have PCD because they've been given a genetic diagnosis, which is actually a non-PCD gene. And Jane's nodding, she gets the same. So maybe in Germany or in the US, but certainly not they quite often from other countries.
Thank you very much, and thank you very much, Hannah, for this presentation. So next, Omran will present an update on the ERN lung registry.
Okay, thanks, Marina, for introducing me. So I will give a little bit of update what happens over time and just see how it moves. Page down. Anything? How can I move the picture? Yeah. This one, I guess. Which one?
Green and then the
okay.
pointer.
Perfect. Thank you. So what you see is we have quite a lot of countries now represented, and I'm very happy here that we actually have now in London, Royal Brompton, Michael Löbinger is here. We have now also an adult group. So we have two independent partners. So we have a pediatric group, Royal Brompton, and an adult group, and I think that's great. We have somebody from Turkey, from Konya University new, and from Lisbon new. So welcome to all those new players. So what about the number of participating centers? You see that this is really growing over time. And you see that the number of people really in the registry is also growing over time. Here you see that especially the new centers, they have not always included all data yet, but you see that quite a lot of centers are also very active. So they really put individual centers, and they are larger centers with more than 300 patients and smaller centers. What you also can see is that we have a variability with the countries. Makes sense depending on the centers. Some countries, like in Germany, we have high numbers, in Britain, but also in Turkey and in Italy and in Denmark. And here you see that the number of genetically defined PCD individuals is really increasing. Also, we have now almost 1,900 individuals with a confirmed genetic diagnosis of PCD in the registry. And I mean, like Hannah was referring to, I could just go back, DNH5 is still, since the discovery we did in 2002, it's still the most frequent gene. So after 25 years, in 2000, we identified the locus, it's still the most prominent gene. So what you see also is that something has changed. When we published in 2002 DNH5 in Nature Genetics, so a long time ago, this was an ODA defect gene. So a hallmark defect gene, and most of the genes in the following years were hallmark defect genes. However, now the group with no hallmark defects is really now going up. So you see now we have approximately 30% with no classical ultrastructural defect, no hallmark defect. That's important. I show here you some data that just came actually online in meta-archive because we felt it's really important. As there was still the PICADA recommended in the old and in the new guideline. I show here you the sensitivity for PCD genetically confirmed individuals. These are 269 individuals that we studied here all had a genetic diagnosis. So many more patients than originally reported for PICADA that were just diagnosed by TAM and video microscopy at that time. So what you see here is that it's really good in identifying laterality defects, 95%. However, even there, 5% of individuals were not identified. If you look in the group with no laterality defect, PICADA had only a sensitivity of 61%. So it's really important. 40% will be not having this threshold. So we also looked in the groups by what is happening, what kind of genetic defect. And here you see that sensitivity for patients that have either an ODA defect or ODA plus IDA or a tubular disorganization defect, you have a sensitivity of 83%. But in the group without hallmark defect, only of less than 60%. And I think it's important because things have changed since the introduction of PICADA. We have more and more patients now with no hallmark defects. So when you look at the individual's questions, I think it's important that you can see that some individual questions, like, for example, neonatal chest symptoms, are really, really good, are still having very good rates. So they are really definitely increased in groups with PCD. So it makes sense, like Amjad was referring to, classical clinical symptoms should make you think about a PCD. So I think it's important, you can also look for hallmark defects, and again, you see that individual questions are good, but you see that the lateral amount of laterality defects, obviously in this group, is much, much lower. That's the reason why PICADA is not doing a good job. So I think it's important for us now, we need a new predictive tool to help clinicians to diagnose PCD. I think this is an important message. It's not the way that PICADA is bad or anything like that. It does not make sense. I think it's important that we know that and that we try to think about how can we make a better job. So recently we published a novel gene responsible for a cystic fibrosis-like disease. It's WFDC2. It's actually a disease where a secreted protein protecting against Pseudomonas infection is mutated. So it's actually a defensinopathy. Looks like CF. You can see here on the right the CF scan. And the interesting part that I noted as a clinician is, they have lots of disease in the upper lung fields. But when we look at our patients with primary ciliary dyskinesia, we have a complete different pattern. We do not have a 5-lobe lung disease in the beginning. We have problems here, and this is a patient with an FEV1 of 30% or even less, 25%, and we will put another email actually around, if people want to participate through ERN Lung PCD core. So most of really important is it's actually a joint study. So it's really that all the people come together. And if you want to do studies like, for example, Eric in Pseudomonas or other people, please go ahead. And we will try always to support everyone. And we are very happy that more and more countries are joining actually the registry. I would like to say that there is just an announcement. So there is the ERN Lung annual board meeting. It's now not in Frankfurt any longer, so it's in Paris, France. So I think it's possibly more beautiful. So people should come and put it in the calendar. And now I would like to come to a point, very important point, and this is we had international ERN Lung PCD core meetings, last in Naples, before in Cyprus. But in parallel, there were also international meetings in the US by the US PCD foundations for many years, actually. And actually, we came together, and Amelia was also instrumental for that at a Gordon Research Conference, where we decided to say that we would like to have one meeting, one annual meeting, where the PCD Foundation, the US PCD Foundation, ERN Lung PCD Core, and BEAT-PCD try to organize one big meeting, and we call it now Global PCD. And there's already also a date I would like to announce here. And I think that's a fantastic news, and please put it in the calendar. And the other things we agreed on, we will not do it all the time in North America. We will circulate. So the next will be in Europe. And obviously, we are very happy that there are also other international people now coming, and we could think about how to go for that. So please put it in the calendar, and I think that's really fantastic that it is such a joint thing. So, and now many, many thanks.
Thank you very much, Heimat. Many good initiatives. And Amelia, you have a question?
No, the registry is amazing. It's a huge, amazing resource to understand more about PCD. I was just going to ask the question about, I think I agree that a new score could be useful. To develop a score, having just learned a lot about these things, you need a population of patients who don't have the disease as well. And so we would need to think about how to get those, because you can't just develop a score on people who already have the disease. It needs to be a group of people who don't.
I like that very much, Amelia, this comment, because I was thinking about that. The score we are using at the moment, the PCD score, the control group was ruled out basically on high-speed video microscopy. I mean, it's not completely, but it was also in this paper stated like that this plays a role. We now published many papers where we have shown that high-speed video microscopy can be completely normal, at least for an expert like me. And so I believe we have a score at the moment where we know that our control group did not work. So I was thinking, what I think it's really important is for this score, we possibly will use all our knowledge. So my idea, I was thinking in my clinics, we have many patients that are sent to me and they were saying it's PCD, but we identified novel diseases that are not PCD, like WFTC2. But we have many patients with PID. We have many patients with CF in our center. So what I think what we will do is we will ask our people here who care also for CF, other genetically defined bronchiectasis disease like PID, etc. And they include those patients, and we will take them as a control group. This is my idea. And this is actually what we want. We want to have a control group that has also bronchiectasis, because we are talking about a chronic bronchiectasis disease. And in this way, we can possibly work on that. This is the best we came up, but if other people are better, I'm very happy about that. But just to take everybody and that somebody just says, oh, you do not have PCD, will not work. So I was thinking to go for positive diagnosis. Susan?
Like Amelia, Haarman, I just wanted to say that the ERN registry is a fantastic resource, but as a representative of Norway, I think I also wanted to add that I was a bit disappointed to see that we had only 2 genetically resolved patients when we have well over 100. So I think it's also important that we present, when there are members of the country in the audience, up-to-date data. There may be other reasons for that.
Did I put here at the moment today no Norway data? I do not know.
I may have misread the slide, but it was just a comment, really, a sort of tongue-in-cheek comment.
I think that's it's an interesting question. And what I can do is, could you go to the first slide, and then I will just go down because I can learn my slides. Where's Norway? Number of PCD per country, number of PCD centers, and it's 2.
Number of PCD individuals per country, we have approaching 130, 40.
Yes, and possibly not all are given into the registry. So I guess that's because this is just a take out of the registry, and possibly Petra can comment better on that. But this is what we actually sometimes experience, is that people are not always giving the data, but we can clarify that.
I know that there are more than 2.
Yeah, okay. Then we will definitely try to clarify, because we would be proud to present higher numbers, Susan. Apologies for that. Yeah, we can try to get better. Okay.
Hi, Haarman, thanks a lot for the very nice talk. And I wanted to actually talk about the genetic diagnosis for PCD patients from Poland, because I can see there are 30 patients in the registry, but as you can see on the next slide, there's only 1 patient that has genetic diagnosis. So whom should I talk to if I would like to clarify that?
I mean, Petra is here, you know, she
okay,
actually made the slides, so I guess that she can help.
Okay.
Sometimes lots of data or patients without any data in the backbone. So we have to work, everybody has to work on filling in the data.
But I like that, because everyone who feels like their way, what we can do is we will now, I think Susan, has a big point, and I like that, and you as well. So we have 2, Susan. No, what I will do now, I will send everyone individually an email. What is at the moment when we look at the data in the registry, and you let us know what you see, and then we can clarify what's going on. I think this is really an important and a good point, and then we can see and learn, because we want to have everyone in, and I'm not sure why this is happening.
Okay, and then the next question is, because you are developing, you would like to develop a new score for PCD, or new diagnostic tool for PCD, but it's based on lung wall changes, so bronchiectasis measurements and localization. What about patients who don't have bronchiectasis?
I completely agree with you, especially if you are young. Let's say you would go at CT scan. It's not the way that this score will be the same for a 75-year-old or a 7-year-old. So if we collect this large data set, we can possibly say, okay, all patients in our registry, more than 3,000 patients, have an abnormal CT scan after following age. And then we can look in each age bin. So when we published our data on FEV1, we published longitudinal data on age bins in ERJ. So we probably have to do the same when we want to do the same score. We have to look in age bins and see what kind of pattern is good. Obviously, in a patient that was just born and has just the situs inversus, obviously it's not a good idea to take chest CT as a good way. So my idea is to look at clinical findings again. so to see whether we can make a better combinations. If it's only clinical, we go for that, but we should also combine it with chest imaging, and we should look at different ages. Yeah, and for the adults, I can definitely say, because I get many referrals from adults with a question, what is the bronchiectasis coming from? And here I can tell you they have usually bronchiectasis. So it's helping.
I agree. This is going to be a very good idea.
But I completely agree with you. It's age dependent.
Yeah, it is age dependent. And then if we would like to improve the diagnosis, age of diagnosis, we of course need to find some objective measures of PCD disease in young patients. But this is going to be very good for people with bronchiectasis. I agree. Thanks.
So I was going to. It was exactly the same as Susanna's saying. Really, is that yes, let's improve on the tools that we've already got, but I'm really concerned at taking our eye off the ball for the. We want to be diagnosing people in infancy, and so CT scans, chest X-rays aren't going to do that. They're fine for the adults who've been missed, but I think we need, as a community, we need to be brainstorming and thinking. How are we going to pick up the patients under the age of 2 when we can actually do something to maybe modify their disease course for the future?
I completely agree with you, Jane. But we have also to face the fact that we have many patients who are 25 years old and they are not diagnosed. So I think we have to put emphasis on all age groups. Yeah.
What I don't want to do is take the ball off the eye. As pediatricians, we want to be picking it up really early. And yes, of course.
Yeah, I completely agree. Yeah. Yeah.
Thanks. Great discussion. We have to stop here, and we have to continue with the next speaker. I would like to invite Marona to present on BEAT-PCD update. Please, Marona.
Can I use this microphone? Thank you.
Yeah.
How does that work?
Yeah, use this one.
Oh, perfect. Every year when I do this annual updates of BEAT-PCD, I think that Amelia gives them to me on purpose because she expects me to fit 40 projects in 10 minutes. And then I feel bad that I leave so many things out or that I talk about some projects in 1 or 2 seconds each. I will try to be very quick, just take this presentation as a highlights of the great work that is being done from this network and all the people that they are involved. And if something seems interesting in you, then just contact us and we will try to bring you in contact with the people who are on these studies so more collaborations can come in. So for anyone who doesn't know what BEAT-PCD is, luckily Hannah covered the ClinGen part, so I will skip that completely since it was so detailed. I will just mention again Celia VAR, which is now at the phase of beta testing and improving the interface so that it will be more user-friendly. I just have a small example in the bottom of how this would look at the user at the time. Hopefully this will be slightly improved and be made even more user-friendly. But this will be an open source database of known disease-causing variants, and people can go in and really see this data and really see if these variants are associated with PCD and what kind of evidence exists for this association. Our patient engagement activities work package is very, very active. I just want to remind to you the Living with PCD study. This study started as COVID PCD during the COVID pandemic, together with patient support organizations all over the world. And with their encouragement and wish to continue participating is now the Living with PCD study, and it addresses many more questions that it's difficult to address through clinical studies. So I have put some of the most recent publications, and we are now organizing a study relaunch with a new name. And we have an ongoing project on social functioning and mental health, hoping to have the next social functioning questionnaire out in the next few months. So if you think that you have patients that they might be interested to participate, please encourage them to check. They can find all the information on the website. And of course, many of them are also informed by their local support group. We also have a great team working on educational material for patients, and the first educational leaflets will be out soon. We are trying for October, which is the PCD Awareness Month, and our study on patient research priorities and participation in PCD research is now at the final revision stage. We have 2 big events. Just to bring your attention on the patient partnership webinar, this is October 8th. This is not just for patients. It has 2 target audience. One is patients who are interested to participate as partners in research, and the other one is researchers and clinicians who want to learn how to engage more in participatory research. So this is coordinated together with ELF, and we invite you all to participate and share your experience or your questions with us. And then we have our annual PCD patient conference, end of November, 29th of November. This has been now the 4th year. It has been quite successful, and we have patients from all over the world, and we try to have at least AI-generated live translations so more and more people can join and understand the discussions. Complete PCD is one of our newer work packages that runs from this last period, and it is an international, aims to be an international bioresource of epithelial cells and samples for PCD research. Many of you might remember that there was a survey circulated, many people from 27 countries participating, telling us that they collect these samples or they're interested to participate in such a bioresource. And now there is a proof of concept study to identify key biomarkers to be used for PCD, especially in trials. And I think for the first one, 6 centers who had already samples and ethics in place have been selected. And there is also a process for ethics for biobanking for future projects, or more centers can be included. We also have completed a systematic review on biomarkers in PCD, which will be presented this Tuesday on the first fully PCD poster session of the European Respiratory Society. So I'm very happy about that we finally have this year, and I hope everyone is there to have a lot of interesting research and discussions. Did I break it? No. The clinical outcome measures work package, there was just so many publications coming out related to this work that I just thought that I will have them on the slide and mention a few. There are, of course, all the papers coming still out of the ProValve study about the natural variability of lung function and the associations between respiratory pathogens and lung function. We have work on pulmonary exacerbations, the study from Cyprus that I think Penelope had presented last year. And also last year, we also had the study on exacerbations from patient-reported information from the Living with PCD study, definition of sinonasal and otological exacerbation as a consensus definition. And I have stolen a figure from Bruna's slide without asking her about barriers of care for PCD. Again, this will be presented and discussed at the same poster session. Very interesting data, so I hope you all can come to discuss this. Another newer work package is this neglected areas for PCD research. And this covers more or less different areas where we have not done so much research in the past. So, of course, upper airways, I feel a bit bad calling it neglected because the last few years we have done quite a lot on that. The EPIC PCD study, we had some discussions in the coffee break with some people. We still recruit new patients and we recruit new centers. So, there is more funding for the study that has been secured and a lot of plans ongoing. We already have 13 centers from 10 countries. 2 new centers recently, and there are 2 more other centers that now finalizing processes of ethics and translating the questionnaires in their local languages for the patients. So the study grows and a lot of new things are coming out. We also planned and have started discussions seeing the need for some more specific guidance for ENT management, a consensus statement for ENT management where there is anyway no evidence specific for PCD. This will be with experts not only from Europe, but also from North America, Australia, to be able maybe to bring together and have some consensus on some topics that I think for ENT disease, there are more disagreements on management than actually for lung disease. So that would be quite an interesting endeavor, and we plan to start soon. We plan to start a study on pregnancy. There will be 2 parallel studies. Bernard Mert is here. He has a plan for a retrospective clinical study using information for pregnant women with PCD about their lung disease and how this went during the pregnancy time and after the pregnancy. And in parallel, we also plan a questionnaire in the framework of the Living with PCD study for the perspective of the PCD mothers who we want them to share their experiences on the physical symptoms, but also the psychological aspects of the pregnancy while living with PCD. And I mentioned social functioning and mental health already earlier. There are many other aspects that we have plans for. Our diagnostic work package is also doing a lot. I don't mention at all the diagnostic guideline. We discussed it. There is this e-training standardizing of video microscopy. This is a big work that has progressed a lot. I think there are some final chapters, maybe Claire can correct me if I'm wrong, that they are now being checked and finalized, and the testing starts end of the year. So this should be out soon too. And there is also the completed systematic review on artificial intelligence in PCD. So the use of digital tools for screening, diagnosis, and disease characterization. And Panagiotis is presenting this poster in this PCD poster session on Tuesday, where I think all of us should be. And this is also a collaboration with the Connect CRC of the European Respiratory Society. They also mentioned the Slack. What is also quite important is that the leaflet that was developed with the task force of the ERS for the nasal and nose standardization, together with ELF, they developed this leaflet for patients. It has been now translated and is available in 9 languages. And there are still some ongoing translations as far as I know, which is very important because patients keep asking us for educational material that they can understand. So we put these resources all on the website, and they're available to be used and to be given to your patients or discussed with them. I will not say anything about the consensus management since Elora had her presentation. I just, of course, want to mention that during the past year, the consensus statement on routine blood testing in PCD, that was a collaboration with ERN Lung, was published, and that was also a huge amount of work. And then we have, if some of you remember, we had said last year that we developed this PCD transition form, that it's quite important to help doctors transition patients with PCD from pediatric to adult care for disease-specific elements. And this is translated already in French and Turkish, and it's tested in some centers in UK and in Paris. And if anyone is interested to access this, translate it, or use it and test it, you can contact us and I will tell you who to contact. I think Guillaume is not here today, but together with Guillaume Thuvenin, together with Amanda Harris, they are leading this process. And last, for the follow PCD patient questionnaire, it is available in 13 languages today, as we finished almost Hebrew now. And we are also, we have ongoing translations for several Indian languages. We have been contacted. I think there are a lot of people who are interested to use this questionnaire because it is quite simple, and they think that they can give it also outside of research to the patients where they're waiting at the examination area, and they have a nice standardized way of collecting patient-reported information on symptoms. So if you're interested to use that again, you can contact me. I'm almost done. We have an amazing early career members network that they are preparing our newsletters. They are in communication with the ERS Assembly 7, the Pediatric Assembly, and now they're doing also a lot of effort to bring information to local pulmonology societies about all the activities in the PCD community. We still organize these expert webinars. We have focused the last 2 webinars on specific projects, and I actually think this is a good opportunity because usually you see events like that. I try to go through 40 projects in 10 minutes. So now we have the chance to have like a webinar of 1 hour discussing a specific project, details, and asking questions. These were the last 2, and the next one is planned for November, where I, together with Vasiliki, who is my PhD student who took over the management of the BEAT-PCD study, we will describe the study and the results that we have so far and our future plans. Just to finish, we already mentioned some of these events. Helmut really advertised the Global PCD Conference. I don't need to say anything else. I mentioned the patient conference. We have a PCD half-day track at the World Bronchiectasis Conference next year in Hanover, which is, again, I think very interesting, and it's a good opportunity. Last time that we did that, it brought a lot of adult pulmonologists who are not so much used to our network, and it was a very good audience to discuss issues about PCD care. And I think that's everything. I just have a slide with our current management committee and some of the people that do all this amazing work, and I would like to thank. And this slide to just show you how you can be in contact with us and find information about our projects. And also we have made a schedule for all the PCD work that is presented at the CRS conference. So this is in our latest newsletter, but you can also scan the QR code on the right to get this. Thank you very much.
Well, thank you, Marona. I was totally blown back. What a lot of project you are heading. It's fantastic. Anyone wants to ask a question or 2? Maybe only 2 questions for Marona before Tom is coming to the stage. Anyone? That was clear. That was clear. Yeah, thank you very much. And Tom, please come closer and present your update on the US. You want this?
From you, please. Thank you. Okay. Again, this is what the 4th or 5th time that I've come to give an update about what we've been doing in North America, and I promise to make this relatively quick because I have the unenviable position of being the person between you and a glass of wine. I can already feel the hostility raising in the room. The other thing is, with regard to this, as you notice, I said this is the final update. I'm not retiring. I'm not dying as far as I know. The NIH funding for this consortium ended. We have 20 years, which is a remarkable run in the United States. But we had 20 years of funding, and so it's now ending, but that's okay. As things end, others begin. Okay, now I'm in the middle one. Thank you. These are just my potential conflicts. PCD Foundation, needless to say, and then a couple of these NIH awards support the research that we are doing in primary ciliary dyskinesia, including the consortium. This is the update. This is the last slide I'm going to show you, which I've shown a few times, of all the various sites that have participated in the consortium across North America. The current sites are Chapel Hill, North Carolina, Bethesda, Maryland at the NHLBI, Washington University in St. Louis, Colorado Children's Hospital and National Jewish Health, University of Washington, Stanford University, and our Canadian colleagues at McGill University and at University of Toronto and Hospital for Sick Children. It's been a remarkable ride. These have been extraordinary people. You can see how many people we've enrolled over the past 20 years. Now over 1,500 individuals were screened. Not all of them had primary ciliary dyskinesia. It was a learning curve. I would say that over 800 of these individuals did ultimately, we found that they had primary ciliary dyskinesia. And again, sounds slightly selfish that I'm kind of focusing on the North American experience, but we could have never have done much of this, either in parallel with our colleagues in Europe and Australia, or more often in partnership. But it's really remarkable if you kind of look back over the past 20 years of where we've gone. You know, I think about the first chapter I wrote on primary ciliary dyskinesia, and I really want to apologize to anyone who read it, because a lot of things that I said was out of ignorance and because I just didn't know. But over the past 20 years, we figured out what we're looking for in primary ciliary dyskinesia as opposed to a child who coughs. We understand an awful lot about this disease starting very early on in life. We've demonstrated the presence of, everyone knew about situs inversus totalis. That's what everyone remembers about primary ciliary dyskinesia from medical school. But the relationship with heterotaxia and other congenital cardiac malformations has arisen over the past 2 decades. We in the United States have focused a certain amount on nasonitric oxide. We've looked at pulmonary function and the acquisition of bacteria in the lungs, trying to characterize this in primary ciliary dyskinesia and across genotypes. We've also, in partnership with so many of our colleagues, discovered PCD-associated genes as well as genotype-phenotype relationships. And one of the important things I'm going to stress at the very end is just remember that not everyone who coughs and has ear disease and has sinus disease has primary ciliary dyskinesia. You do not want to miss people who have primary immunodeficiencies, because that is a deadly mistake. We partner with the PCD Foundation, which is an extraordinary organization of parents and affected individuals with PCD. And we've also partnership with the Immune Deficiency Foundation during the past 5 years. And we've been involved in a number of clinical trials, some of which have been described earlier today. With regard to the consortium, our 3 final projects that we focused on was trying to understand the genetic etiology of suppurative airway disease, and specifically bronchiectasis. And we're trying to understand what are the similarities, but perhaps more importantly, what are the differences between PCD and immune deficiencies? Are there clues that you can pick up on the physical examination, on the history, on the testing, that tells you this is more likely to be an immune defect versus primary ciliary dyskinesia? We enrolled 236 out of the 300 that we planned. There's a little thing called the pandemic that got in the way for the first couple of years when we started, and all 8 of the sites participated in this. We also discovered that we had a large number of individuals who we could not make the diagnosis using conventional diagnostic panels or even exome sequencing. And so we're partnering with the National Institute of Allergy and Infectious Diseases, where we're going to be doing whole genome sequencing in individuals who are enrolled in the 5906 study, where we do not have a clear genetic diagnosis. And so we have a number of those individuals that have already been sent samples, and in some instances, individuals have been sent to Bethesda for further genetic testing, but also laboratory confirmation of an immune defect, because these are the guys who know how to do that. The second project, 5907, was looking specifically at pulmonary exacerbations. It was a longitudinal observational study, and that we enrolled patients over 13 months, where we are looking at their clinical features. We're looking at home spirometry. We're looking at mycobacterial assays and inflammatory markers in sputum samples and in nasal samples that were collected from these individuals, trying to pin down some physiologic correlates with what we think is a pulmonary exacerbation. And we're really hoping that this is going to provide some guidance, especially as we're doing future clinical studies. 6 of the sites participated. actually take that back. 7 of our sites participated. We enrolled 105 out of 125 planned. And then the last project was led by Adam Kimple, who is a pediatric otolaryngologist in Chapel Hill, and he, with 4 other sites, basically did a cross-sectional study trying to define the upper respiratory manifestations of primary ciliary dyskinesia, looking at the clinical features, looking at the appearance of the mucosa, as well as collecting samples from the nasopharynx to do microbiological and inflammatory assays. One thing that we noticed, and I'm glad Hamid had brought up the SNOT-22, because then I didn't have to say it myself, but basically it's a measure of nasal symptoms in individuals. And he was able to, Adam was able to demonstrate pretty clearly that there is a real difference in terms of symptomatology between people with primary ciliary dyskinesia and those who have inborn errors of immunity. And I suspect we're going to find similar differences when we start looking at the biochemical and the microbiological aspects of the nose. I just want to highlight 4 papers that are in, or 4 studies that are in various stages of review or potentially publication. This is a study that was initiated by McGee several years ago, looking at nasal nitric oxide and specifically tidal breathing, which was something that came up as part of the guidelines. We actually had the data. I wish we had published it before. But the bottom line is that yes, you can do. We found that using a chemiluminescent analyzer, that you can do tidal breathing to detect primary ciliary dyskinesia in children as young as 1 year of age. The important aspect of this is, the way we approach this, it was a very standardized approach that all the consortia did it the exact same way. And so when we did the initial study to determine what the cutoffs would be, then we were able to validate at other centers that were using the exact same technique to show that it was also accurate, though it did not identify everyone with primary ciliary dyskinesia. This is a study that was led by Brie Kinghorn. She's been working with our colleagues at Erasmus University using an automated CT imaging. And there's a lot of data in this paper that is yet to be published. But one of the interesting aspects of this that I believe Dr. Chalmers had brought up regarding mucus plugging is that mucus plugging may be a really interesting phenotype or radiographic phenotype in primary ciliary dyskinesia. And it may be telling us a lot more about what's happening in the smallest airways. And what Brie was able to show, in addition to an awful lot of data, was that individuals who had, that people with, that the percent of mucus plugging increased over time in children with primary ciliary dyskinesia, but more specifically, people who had the interdining arm microtubular derangement defect, CCDC39, CCDC40, had it much greater, which correlates with what us and many others have shown in pulmonary function, let's just say decline in those individuals. One of the other interesting aspects of this is that we used mucociliary clearance assays in vivo in individuals with primary ciliary dyskinesia. And Larry Ostrowski, also at University of North Carolina, Chapel Hill, found something rather interesting, that he didn't look at all people with primary ciliary dyskinesia. He looked at individuals who had pathogenic variants in either DNH5 and RSPH1, because there's some evidence in the literature that maybe RSPH1 has a milder respiratory phenotype. And what we found, or what Larry found was that if you're looking at mucociliary clearance, it's not there. It's pretty much non-existent, pretty much confirming data that Kim had published a few years ago. But if you look at cough clearance, there is a difference between individuals DNH5 and RSPH1, such that the cough clearance in these individuals were pretty similar to what we saw in the normal volunteers. So is this telling us there's something different about the smaller airways in those patients? Is it telling us something that there is some residual function of this whirling cilium in individuals with radial spoke defects? I do not know, but this is something that we're definitely going to follow up on. And then the other thing, and if you want the best reason that I have to come and give this talk, is that a couple of years ago, I was showing the number of individuals that we had identified who had inborn errors of immunity that were sent to us for evaluation for primary ciliary dyskinesia. And about a week afterwards, Debbie sent me a note says, I got people with that defect that you showed that has this. And so we basically put our data together, and sent it in as a publication, and the Journal of Pediatrics accepted it. I guess I shouldn't sound so surprised, but I am. But one of the important things, and I want to stress this, I cannot stress this enough, this syndrome, this defect, there's a treatment. An FDA, I don't know whether Europe, but there's an FDA-approved treatment for this disease or for this defect. If you miss this, or if you mistake these people as having primary ciliary dyskinesia, they're not going to get the treatment, and they could potentially have consequences as a result, which can include malignancy and early death. So I told you something new is coming down the road. So miraculously, in the current National Institutes of Health environment, we got a grant funded. And when I say we, Rick Boucher and Ken Olivier, where they put together a rare bronchiectasis disease consortium, looking at individuals who have bronchiectasis across 4 rare diseases: primary ciliary dyskinesia, humor immunodeficiency, severe alpha-1 antitrypsin deficiency, and STAT3 hyper IgE syndrome, previously known as Job syndrome. 4 sites are involved in this. This is the other equally miraculous thing about this. 4 sites were funded. 2 of them are not in the United States. The Royal College of Surgeons in Ireland and McGill University. But nevertheless, we got funded. We're going to do the work. And specifically, we have 2 projects. One is looking at the longitudinal study of outcome measures and biomarkers in rare bronchiectasis disease, and we're using a number of different modalities to test this. And we're looking also at rare bronchiectasis disease markers and novel treatable traits, specifically focusing on the smallest airways in people with these diseases. The last thing I want to say, and I have to thank a number of people in the room because you have volunteered or maybe were coerced into serving as reviewers for our development of a network for clinical trials, not unlike what Kim has been leading for the past few years. And a few years ago, we don't think we could have done this because we didn't have nearly enough sites. We didn't have nearly enough centers with the expertise. But as you can see from the map, we have a number of programs that have gone across North America that have become very much involved in primary ciliary dyskinesia. They use a standardized approach toward diagnosis. They use a standardized approach toward monitoring these individuals and to some extent the management of people with primary ciliary dyskinesia. And so we've opened this up to start to develop a network of centers that would be specialized in clinical research as well as clinical trials in the United States. We received 20 applications. A couple have been withdrawn since then, but there will be a review in October, and we should be able to inform the sites that are participating in this shortly. And then finally, I have to kind of highlight the people who did all the work. These are all of our colleagues. This is just a very small number of individuals that participated in the consortium over the past 20 years. But I cannot say nearly enough about the PCD Foundation, particularly Michelle Mannion. The PCD Foundation was formed about 6 months before we started the consortium. And she has been our greatest champion and has been just remarkable in her support in what we have done and has been as proud as anybody as to where we have collectively gone. And so with that, I'm done. Let's go get some wine.
Thank you very much.
No questions? Tom?
No problem. No problem. Thank you, Tom.
Just before everyone stands up and leave, before saying thank you everyone for coming here today and listening and participating, we have an award to give. Yes, yes. Last year, for the first time, I think we gave this award for the best presentation from our early career member session. So we had some people who judged, and we are very thankful too. And this year, the award goes to Joe. Thank you very much for your presentation, Joe. Thank you very much for this very lively and excellent Do you want to say anything? Then thank you very much. Let's go and enjoy some wine and see you all next year in Montreal.